Welcome to Ace My Exams Learning. Let us get started with today's learning.
A section of a DNA molecule has the following base sequence: C, T, T, A, C, A. Question. Name the nitrogenous base represented by C in the DNA molecule. The nitrogenous base represented by C in the DNA molecule is cytosine. DNA, deoxyribonucleic acid, is composed of four nitrogenous bases: adenine, A, thymine, T, cytosine, C, and guanine, G. These bases pair in a specific way, forming the structure of DNA. Cytosine, C, always pairs with guanine, G, through 3 hydrogen bonds. This complementary base pairing ensures the stability of the DNA molecule and allows for accurate replication during cell division. Question. The percentage of guanine in this DNA molecule is 30%. Give the percentage of thymine in the same molecule. The composition of DNA follows Chargaff's rule, which states that 1. The amount of guanine G always equals the amount of cytosine C. 2. The amount of adenine A always equals the amount of thymine T. 3. The total percentage of G plus C plus A plus T in DNA must add up to 100%. Therefore, given that guanine G equals 30%, and since G pairs with C, the percentage of cytosine C equals 30%. Hence the combined percentage of G and C equals 30% plus 30% equals 60%. This means the remaining 40% is for adenine, A, and thymine T. Since A equals T, we divide the remaining 40% by 2. Thymine T equals 40% divided by 2 equals 20%. Question. Give the mRNA sequence, from left to right, for this segment of DNA C, T, T, A, C, A. The process of converting DNA into mRNA is called transcription. During transcription, mRNA is synthesized using DNA as a template. However, in RNA, the base uracil U replaces thymine T. The complementary base pairing rules for transcription are: 1. Cytosine C pairs with guanine G. 2. Thymine T pairs with adenine A. 3. Adenine A pairs with uracil U. Let's apply the rules to DNA segment C, T, T, A, C, A. C pairs with G. T pairs with A. T pairs with A. A pairs with U. C pairs with G. A pairs with U. Therefore, the mRNA sequence for the DNA segment C, T, T, A, C, A is G, A, U, G, U. Question. The table below shows the DNA triplets that code for some amino acids. Give the sequence of amino acids that would be coded for by the section of DNA above. DNA contains triplets, groups of 3 bases, that serve as the genetic code for amino acids. Each triplet corresponds to a specific amino acid, as determined by the given reference table. Given the DNA sequence, C, T, T, A, C, A, we divide it into triplets and become C, T, T and A, C, A. Next, we match each DNA triplet to its corresponding amino acid using the provided table. Therefore, DNA triplet CTT matches amino acid glutamic acid and DNA triplet ACA matches amino acid cysteine. Therefore, the sequence of amino acids that would be coded for by the section of DNA above is glutamic acid, cysteine. Question. Give the anticodon for the amino acid methionine. To answer this question, we start by identifying the DNA code for methionine. Each amino acid is coded by a specific 3-letter DNA sequence, known as a triplet. The DNA triplet that codes for methionine is TAC. Next, we use the base pairing rules to convert DNA to mRNA. T as DNA becomes A when converted to mRNA. A as DNA becomes U when converted to mRNA. C as DNA becomes G when converted to mRNA. Thus, the DNA triplet TAC is transcribed into the mRNA codon A, U, G. To find the tRNA anticodon for the mRNA codon A, U, G, we match the bases using these rules to pair mRNA and tRNA. The base A, AID9, in the mRNA codon pairs with U, uracil, in the tRNA anticodon. The base U, uracil, in the mRNA codon pairs with A, AID9, in the tRNA anticodon. The base G, guanine, in the mRNA codon pairs with C, cytosine, in the tRNA anticodon. Therefore, the base A in A, U, G pairs with U, U pairs with A, and G pairs with C. Therefore, the tRNA anticodon that corresponds to the mRNA codon A, U, G is U, AC. This anticodon ensures that the amino acid methionine is correctly placed in the growing protein chain during translation. Question: describe how a mutation on DNA may change the structure of a protein. A mutation in DNA alters the genetic code, leading to changes in the mRNA formed during transcription. This results in different codons in the mRNA sequence. These altered codons match different tRNA molecules, which bring different amino acids to the ribosome. As a result, the protein produced may have a different amino acid sequence, causing it to fold incorrectly and potentially lose its function. This disruption in protein structure can lead to non-functional proteins or altered functions, affecting the overall function of the cell.
To access more learning and exam preparation materials, go to www.acemyexams.coza. This link is also in the video description below. Thank you for watching this video. Remember to subscribe and be the first to know when we upload new videos.